The fascinating thing is that like a lot of organisms have mechanisms for preventing like DNA damage. So if you, for example, so I was in the epigenetic clock field for a while, and maybe we'll talk about this later. But the fascinating thing is, when you look at epigenetic clocks and how they measure aging, you can create an epigenetic clock that measures aging consistently across tissues and cell types. That was like Steve Horvath's big finding in 2013. But if you take his clock and you apply it to sperm cells, it's always off. And it's always the sperm cells either don't seem to be aging or aging like 70% slower than every other cell type.
This is something common with like germline cells, right? Or okay, yeah.
Yeah, exactly. And so like oocytes have like similar things, although oocytes sort of cheat a little bit. And I think they just like, there's a really cool paper that showed they just like turn off part of the electrode transport chain. And so they kind of like get into this hibernation stage. But sperm don't get to do that. They have to keep dividing. They have to stay alive. They have to stay metabolically active. And yet their genomes don't seem to age nearly as much as the other cell types and tissues. Kind of escaping humans a little bit. There's these deep sea vent bacteria that live in temperatures that are crazy high and very damaging to DNA, and they create small molecules that protect the genome. Of course, there's a naked mole rat story too, where they have like these I think long lipids that protect DNA from damage. And so it's not like an unthinkable idea. We're not building something that has never existed before. I think we are just taking the best parts of everything and seeing what we can translate into humans.
Today, I'll be talking to Alan Tomusiak. He has a PhD in the biology of aging from the USC Buck Institute and is currently founder and CEO of Permanence Bio, a longevity startup developing genome stabilizing therapeutics. Today, we'll be talking about DNA protection mechanisms, why preventing somatic mutations will be extremely important for longevity treatments, and much more. Welcome to the podcast, Alan.
Thank you. It's amazing to be here.
So I think my first question is to set like the broad picture of what we'll be talking about today. One of the theses for the field of longevity at large you seem to be really interested in is that protecting the genome is of far higher importance than almost any intervention we can do. You have a really great blog post that walks through like all the reasons you believe this, which I'll attach in the description of this video, but I'd love for you to just give that as like a recapitulation.
Yeah, I think on a high level, there's no such thing as a more important or less important part of aging. I think almost any biologist who's working in the field of aging will tell you that all of the hallmarks of aging exist. I think the one dimension where I think genome stability has a certain degree of primacy is in the question of like, what is the order of events, right? So you have different hallmarks of aging. You have extracellular matrix breakdown, mitochondrial breakdown, genome instability, epigenetic alterations. And the question is more, it's not so much like which of these is most important to aging, but which happens first, right? Like when you're 20 and 30, what are the kind of first things that go wrong that lead to the other things? What is the sequentialism here? And so in that case, it seems to me, or from everything that I've seen, that genome stability seems to be the first thing that breaks down. And there's a few different areas from which you're going to look at this. Like, for example, one Twitter thread that I want to write at some point is looking at every single hallmark of aging and how a genetic defect in each hallmark of aging, to what extent does it look like aging? So for example, there are people out there who have a mutation in elastin. And so you can ask like, okay, if you have a mutation in elastin, how much does that look like aging? And of all the sort of progerias, the accelerated aging disorders, every single one of them has a mutation in some DNA repair or some genomic maintenance gene. So I think that's kind of like one of how I think about it. I think about it less in terms of what is the most important. I think about it in terms of what is the causal relationship and what is the most causal upstream factor for aging.
And so, yeah, so I guess like when I say important, I do mean like there's this like base layer upon which all the other hallmarks of aging seem to derive some, they have some sort of like causal of like, if you have some somatic mutation, it causes this hallmark of aging. I was like, is it correct to think of it that way, where somatic mutations genuinely are the cause for like the 6 other hallmarks?
I'm very careful about saying genome stability. So somatic mutations on their own, it's a very controversial field to what extent somatic mutations specifically. So like when I'm saying mutations, like a change in the DNA code versus damage happening to DNA and then all the downstream effects of that. So as an example, the somatic mutation field used to be very, like, I think one of the most dominant theories of aging was that it was caused by mutations. More recently, there's been a lot of work showing that there's individuals out there with certain mutations in a polymerase enzyme that leads to far higher rates of somatic mutations. They do not look like they have accelerated aging. Specifically, I think it's polymerase epsilon, and they have like a 15 times higher basal somatic mutation rate in the colon. And like, sure enough, like they get a lot of cancer,
Mm.
but they don't age faster. And so there's still ways that you can think around this. Like I wouldn't say the somatic mutation theory of aging is dead necessarily. Like there's people who, there's certain papers that are really compelling showing there's like selective mechanisms, like in clonal hematopoiesis, you have an advantage of certain somatic mutations that then leads to aging-like phenotypes. There's recently a very fascinating paper that came out looking at chronic kidney disease. And in mice, they show that somatic mutations lead to defects in, I want to say, like the lamina A gene that then leads to sort of like dysfunctional senescent cells that look a lot like their aging cells.
Mm-hmm.
But I think overall, the state of the evidence right now doesn't quite support the fact that somatic mutations are the cause of aging. I would say it's one factor upstream. Like the way they think about it is that you have DNA damage that then leads to somatic mutations, which leads to cancer. But you have DNA damage that leads to other things, including epigenetic loss of information, and that is more concretely an aging phenotype.
Mm-hmm. I guess like one thing the one potential mistake I've been making is I'm equating genome instability with somatic mutations. Is that incorrect? And there's actually some nuance there?
There is a lot of nuance.
Okay, yeah.
And I'm kind of thinking like, is there a way to say genome instability? Like, is there a way to like rephrase that? Because a lot of people will say, oh, like genome instability, mutations. But genome instability means a lot of things. I mean, even on a meta level, like thinking about the 3D architecture of the DNA, right? Like that changes so much of how, senescent cells will just like throw out DNA, right? And that is definitely genetic instability. That's not a mutation.
Like throw out DNA from itself?
From the nucleus into the cytosol.
Oh, strange.
Which is very weird. And it's not sure whether it's like a controlled process or not. But telomere attrition is a form of genome instability in its own way. DNA, like you can think about abasic sites, crosslinks, double-strand breaks, like all of these things are derived from genome instability, but they're not mutations.
Yeah.
So I take it one step higher level than looking at mutations specifically.
Okay.
I do care a lot about mutations. I think mutations are incredibly important from the point of view of preventing cancer. And so for kind of what I'm working on right now, to some extent, like mutations are a good enough proxy for genome instability, because at the end of the day, like genome instability, mutations both cause cancer very directly. Therefore, prevent one, you're fine. But I think if we're thinking a bigger level in terms of aging, it's not quite somatic mutations. I think it's one step higher.
Okay. And so the primary lens by which at least like Permanence is working on is like trying to prevent somatic mutations from occurring in the first place.
I would say so, yeah. Of course, like if you can prevent other types of DNA damage that are not, like as an example, like one form of genome instability, one thing that comes from genome instability is inflammation. Like a cell picks up the fact that genome is damaged, and then it releases all sorts of different factors that lead to aging, but also cancer creation. And so, like I would say, mutations are a very, very useful readout for us, specifically because it's also relatively easy to measure, and it's like a kind of a binary thing. Like you have a mutation or you don't have a mutation. Like how many mutations do you have? And it's relatively easy to interpret. If you protect the genome, then you shouldn't be getting as many mutations. But I would say it's one of many things that we care about.
Okay. And so for Permanence, you guys are building chemical treatments to prevent the genome from undergoing mutation. Could you walk me through what the general lines of like medicinal chemist reasoning you're using when developing these sort of things? Like what are the primary directions you've historically seen in the field, and where do you think the field is going?
Yeah, that's a very good question. And where I think where the field is going, or has been going, is finding ways to boost DNA repair.
Okay.
I think that's by far like the most common thing. And so what people have looked at is they found all sorts of different sort of natural compounds that improve DNA repair. Certain drugs that seem to have increase the fidelity of DNA repair. I think that's been by far the most common way in which people have kind of gone at the problem. We're doing things a little bit differently in that we're trying to actually prevent the damage. So instead of trying to improve repair, we can see like, can we just stop the DNA from being damaged originally? And in my mind, again, this is like kind of going down this like key theme of how upstream can you go? And the most upstream way of preventing genome instability is to prevent the damage in the first place.
Yeah. It feels like at least some of the sirtuin stuff was concerned with trying to boost natural DNA repair mechanisms, and it doesn't seem like it actually like worked out particularly well. Is that like, one, is that correct? And two, why do you think it is the case that boosting existing DNA repair mechanisms haven't empirically worked all that well? Or have they actually worked well?
Yeah, that's a very good question. Yeah, there's a lot of interesting work in this area. Like, there's Sirtuin's story is definitely a compelling one. Most recently, the Dream Complex story out of Bjorn Schumacher's lab is very, very exciting. Um, I think the main reason why the biggest challenge for everyone building in that field is that DNA repair is so intrinsically tied to cell cycle state
Mhm.
or cell cycle arrest. Like, if a cell knows that it is about to, you know, divide a lot, then there are certain mechanisms that it turns on in terms of DNA repair and certain ones that it turns off. And if you're boosting DNA repair a lot, that is a pretty clear signal to a cell that it should probably not be dividing because clearly there's damage happening.
Mhm.
And so I think there's been a lot of, there's this, the most directly, I would say, like outside of the kind of like therapeutics realm, is looking at P53 mice, where if you turn up P53 in mice, the idea was, oh, you prevent cancer because elephants have like 7 copies or whatever. And the result is that these mice are actually less healthy. Like, they do get less cancer, but they have, to my understanding, hematological deficiencies because their cells that should be dividing a lot are not dividing because they're recognizing, oh crap, there's a danger signal here,
Mhm.
right? And that's just like across the board, no matter what DNA repair kind of complex you look at, there is definitely like some tie to cell cycle state and stress state.
Mhm.
I think that's the main reason why it's been tricky.
Mhm.
I don't think that it's impossible. Like, for example, a number of companies now are thinking about, oh, well, if you look at cells that are already out of the cell cycle, right? Like, what if you specifically boost DNA repair in neurons?
Yeah.
Like, would that work? And I think that's pretty interesting. We haven't seen a big success yet, but we very well could.
Okay. And so, but you're going the opposite, well, not the opposite route, but the route of trying to prevent the mutations from happening in the first place rather than fix mutations have already occurred.
Precisely.
What's your mental model for thinking, like, what does protection actually look like?
Yeah, I mean, the fascinating thing is that like a lot of organisms have mechanisms for preventing like DNA damage. So if you, for example, so I was in the epigenetic clock field for a while, and maybe we'll talk about this
Mhm.
later. But the fascinating thing is when you look at epigenetic clocks and how they measure aging, they measure age. You can create an epigenetic clock that measures aging consistently across tissues and cell types. That was like Steve Horvath's big finding in 2013.
Mhm.
But if you take his clock and you apply it to sperm cells, it's always off. And it's always the sperm cells either don't seem to be aging or aging like 70% slower than every other cell type.
This is something common with like germline cells, right? Or
Yeah.
okay, yeah.
Yeah, exactly. And so like oocytes have like similar things, although oocytes sort of cheat a little bit. And I think they just like, there's a really cool paper that showed they just like turn off part of the electrode transport chain. And so they kind of like get into this hibernation stage.
Mhm.
But sperm don't get to do that. They have to keep dividing. They have to stay alive. They have to stay metabolically active. And yet their genomes don't seem to age nearly as much as the other cell types and tissues. Kind of escaping humans a little bit. There's these deep sea vent bacteria that live in temperatures that are crazy, crazy high and very damaging to DNA.
Mhm.
And they create small molecules that protect the genome.
Mhm.
Of course, there's a naked mole rat story too, where they have like these, like, I think, long lipids that protect DNA from damage.
Mhm.
And so it's not like an unthinkable idea. We're not building something that has never existed
Mhm.
before. I think we are just taking the best parts of everything and seeing what we can translate into humans.
I guess I like to like create like a mental model of the system. You have this like nucleotide and like phosphate backbone, and you have like potentially like reactive oxygen species like kicking nucleotides out. You have like background radiation of the universe like damaging the nucleotides. Like, what is like these like phospholipids and naked mole rats or any of these other DNA protection mechanisms? What is it physically doing to the DNA to prevent the damage from occurring in the first place?
It depends. So there's a whole spectrum. So some of them don't interact with DNA at all. Like, a lot of these compounds are just like basically very powerful antioxidants that
Okay.
exist inside of a cell. So that the downside is like those are very powerful. The downside is that you need a very high concentration of them to do anything. You can imagine a cell is very big, and the amount of distance that a raw species takes to actually hit the DNA is very, very tiny.
Mhm.
And so you have to be either very lucky or you have to have a very high concentration of antioxidant to see an effect.
Mhm.
Or you have to localize it to the right place. Some kind of compounds are very directly DNA binding, so they will change the 3D structure of the DNA in order to make it more resilient against damage.
Hmm.
Or like they'll compact it in some ways. And again, you kind of need high concentrations of compound for that. But for a lot of these, especially bacteria, that's kind of the interesting mechanism.
Is potentially a dumb question. If DNA is compacted into the chromatin is it more resistant to damage or less resistant to
It's
damage?
more resistant.
Okay.
Yeah.
I guess that makes some intuitive sense. Um, and so on one hand, you could perhaps build like better antioxidants. On the other hand, potentially you find some ways to like stabilize the phosphate backbone, like interact with the chromatin in some way. Like what is the primary axis of variation in the system that you think is the most important thing to like poke at?
I see. Yeah, when I was thinking about this, so this is a very complicated answer. There's kind of I'm looking at it from both a top-down and a bottom-up angle. The top-down angle is not even thinking scientifically, but thinking about indications. So, for
Okay.
example, like if you have a human disease, like which human disease can you treat such that like that would be effective and as where a genome stabilizing compound would be effective. That's kind of what landed me on cancer prevention.
Mhm.
And that cancer is, we can have, it's a very debated topic to what extent genome stability is relevant to aging. Of course, like I'm on the side that's very, very important. But in cancer, it's not disputed. Cancer seems to be largely driven by mutations and inflammation. And so if you could prevent mutations, then there's a lot of cancers that you can stop almost entirely.
Mhm.
And so that became a little bit of a no-brainer. Plus, I mean, the just the sheer, impact that you could have on people by modulating DNA repair, improving it for cancer prevention is huge. And so we're kind of like driven by that as well.
When you're developing DNA protection molecules, how are you actually testing that whatever protection you're conferring to the molecule, it actually works? Is it just you do really like deep sequencing of single cells, or is there some other proxy measurement you're using?
Everything. Yeah. So this is, I think, where Permanence Bio is really stands out. And like what we're doing is very special. I think there's a lot of the biggest challenge in the genome stability DNA repair space has been figuring out like how do you screen? How do you test things, right? Like if you just blast cells with an enormous amount of radiation and see which ones survive, the hits that you get are not DNA repair related. They're like hormesis proteins that like sort of shut down the cell and protect it from doing anything, like completely shut down cell cycle, completely shut down everything. And so how do you tune an assay to tell you the genome protection mechanism, but not anything else? And then there's ways to like mess it up. Like people look at gamma H2AX a lot. And for, listeners who don't know about gamma H2AX, it's like when there's a double strand break, there's a little marker on a histone nearby where the cell tags it. And that tag is a sign that, hey, there's damage that's happening here. And that recruits a ton of DNA repair machinery to get to that area. We screened a whole bunch of compounds at the beginning. We're basically trying to compare the things that we're developing versus the best in class, that other researchers have found. And we found that some of the best DNA repair molecules are not only not hits, but they actually increase gamma H2AX signal. So what I mean by that is like nicotinamide, like very well known to be a very good DNA repair booster. I think canonically accepted as such, and we see a 20% increase in gamma H2AX signaling after we induce damage in a cell. And so what you'd immediately think is, oh, that's terrible. That means that you're actually increasing DNA damage, but you're not. You're actually improving signaling. And that's also a good thing. And so for us, kind of like the long way around to getting to your question is that we're doing a lot of things. Like gamma H2AX is one of them, because if we can actually reduce the damage, then we reduce the damage signaling. And therefore that's good. But we're also looking at sort of broader things. So if we have a kind of a, we're experimenting with hematopoietic stem cells. They're very genomically unstable. In the bone marrow, there's a very small percentage of oxygen. And so that's very easy for them to exist. As soon as you take them out of the bone marrow, suddenly they're exposed to a ton of oxygen. So the genome instability, jumps up. And so if you have a compound that can, for example, improve genome instability in hematopoietic stem cells, they should be able to grow better. You should be able to see a whole bunch of different flow markers that indicate the cell is in a healthier state, and those should, remain high even as the cells are dividing in these like unnaturally high oxygen conditions. And there's also a diversity of insults that you can do. People like radiation a lot because that's fairly direct, but of course, the type of damage that you're seeing with radiation is not the type of damage that you see with formaldehyde, which is not the type of damage that you see with H2O2, which is not ENU. So what we're trying to do is a whole battery of things at the same time, and then see if we can get hits in everything.
I was going to ask, is there a way to discretize different types of DNA damage? Or is it kind of like all in the spectrum and it's hard to categorize what is what?
This is also a very good question. So I think there's two ways to do it. One is you have insults that should be causing mostly one type of damage. And there's very good papers that have sort of like mapped out every single type of DNA damaging drug and roughly what type of DNA repair mechanism it's impacting. And so you can just screen across, let's say 6 or 7 insults and say, hey, which one of these 7 is our compound most effective against? The only downside to that approach is that a lot of these will not be very specific. So some will like damage one, but also another mechanism. But it's very efficient. The other way they can go about it is you have some cells and then you knock out their ability to repair one type of damage. And then you cause a just general damage. And then you see if, to what extent can your compounds rescue those particular cell lines. I think both approaches are good. We're more leaning on the first.
Is there a particular cell type that you're most interested, or like at least like day to day, you're most interested in working on? Or is Permanence Bio really like spread across many different cell types?
It has to be many different cell types. Yeah, especially for cancer prevention. DNA damage, what I've noticed is that there's a big distinction between how slow dividing cells, non-dividing cells, and fast dividing cells deal with DNA damage. So as an example, I mentioned earlier that there's these, there's people out there with a disease where they have a mutation in a polymerase that then leads to a 15 times higher mutational burden. And then you mostly see that mutational burden in the colon. And very fast dividing cells, they primarily have very high rates of colorectal cancer. In contrast, if you mess up another genetic pathway that leads to DNA repair in humans, that leads to cocaine syndrome, they don't get colorectal cancer at all. Instead, it's a major neurological disorder. These are mostly impacting non-dividing cells. So it's mostly autoimmune and neurological. Werner syndrome impacts a third batch of DNA repair. And there you see like slow dividing cells get messed up. So these patients have like, something that looks a lot like diabetes. Their skin starts to like, kind of like look wrinkled. And it's the most physiologically accelerated aging looking model that I've ever seen. So we need to be effective against many different cell types and many different types of damage.
How should I think about DNA damage, not in the context of like environmental insults, but in inherent genetic defects in an individual? So like the example that I'm thinking of is like someone has a BRCA mutation, they're unable to repair, I think like double DNA strand breaks. Do you imagine something like whatever Permanence Bio is like amenable to fixing that problem? Or if there is like a genetic defect that is as a result causing DNA damage, that's a little bit trickier of a problem to fix.
No, and it's actually very, we think about that a lot, mostly because when we think about clinical strategy. So if we want to go big picture cancer prevention, to run a cancer prevention trial in the general human population without stratifying populations at all, that's, I think, somewhere north of $100 million and 5 years and tens of thousands of patients. So looking at individuals who have a high risk of cancer is absolutely something that we're deeply interested in. And there's sort of like gradations here. So you mentioned BRCA carriers have a much higher risk of breast cancer. We also, have thought a lot about Lynch syndrome, where these patients have a 70% lifetime risk of colorectal cancer. So that's an even higher risk of cancer. And then there's like sort of more rare / ultra rare diseases like Fanconi anemia, where they're guaranteed to have cancer. And so kind of as we're thinking about our path to clinic, we might want to start, we'll very likely want to start at these like higher risk patients, and then gradually move on to higher, lower risk patients to prevent cancer in them as well.
I guess like the larger question is like, are there some forms of DNA damage that aren't amenable to these protection mechanisms, or are all DNA forms of DNA damage amenable to DNA protection?
I mean, in theory, you can only get DNA damage by virtue of having some insult that causes DNA damage. Some are trickier than others. The main one that we think a lot about, kind of from the point of view of this question, is replication stress, right? Is replication stress inevitable? Or, I mean, inevitable is a strong word. Is it purely dependent on polymerase fidelity, right? So if it's only polymerases that can cause DNA, that are the contributor to replication-mediated DNA damage, then like DNA damage protection mechanisms are not going to do a whole lot. It's going to have to be, at that point, some sort of gene therapy or some sort of interesting drug that boosts DNA polymerase fidelity. But aside from that, no, I think DNA damage is pretty central and will impact everything.
There are some scenarios in which like a certain drug modality is like learning some aspect about a certain drug modality is fine to do in an in vitro setting because it transfers pretty well to in vivo settings. I think like one common example people use is that you don't typically need to worry too much about antibody toxicity, so it's fine to test it in vitro. it'll probably translate in vivo. With DNA repair mechanisms, like what is the situation like there? Is in vitro like almost like usually perfectly fine to like test everything in and you expect that'll translate pretty well? Or you are primarily testing in animals and even then there's like worries of like cross species that might not work.
Yeah, that's a very good question. I think you learn a lot from cells.
Mhm.
So as an example, especially from the point of view of things that could go wrong. So things that could go wrong with DNA repair, a lot of them have to do with cell cycle rest, kind of like I mentioned. Like there's this like deep entrenched talk between not replicating and doing DNA repair. And you can do experiments and rapidly dividing cells, activated T cells, hematopoietic stem cells, like you name it, in a cell culture model, and that will be really, really informative as to whether or not you'll see that kind of problem in the mouse. And so there are a lot of things that are going to be helpful. Others, less so. I mean, my big questions are kind of like the ones I asked before. Are there certain types of cancer that are going to be more amenable towards treatment than others? That's hard to tell unless you test against every single type of cell in an in vitro model. So
I've
yeah.
always been a little bit curious and like again, like potentially very, very dumb question. How well does like in vitro cancer mirror real life in vivo cancer? Like what are the big distinctions there? Like what are you not accounting for? What are you accounting for?
The immune system is the big thing that you miss.
Yeah, that's fair.
That's the toughest one.
Mhm.
And I kind of mentioned before, like cancer is, I think, largely described by two things: inflammation and mutations. And by doing an in vitro model, unless you have a very sophisticated organoid model, you're missing the inflammation part.
But like, does the immune system still matter for the problem of DNA damage protection?
Oh, absolutely.
Oh.
Yeah. I mean, there's even cases where let's say that you don't prevent mutations at all, but you modulate the immune system's response and then you have a huge effect. I mean, one thing that people, even I thought a lot about is autoimmune disorders. Like a lot of those are driven by genome instability, but they're not driven by genome instability because the cells themselves are dysfunctional, but they're theorized to come from the cells sending out danger signals to the immune system, which then comes in and floods and causes chronic inflammation. And so like the actual DNA damage, mutations barely matter. It's just this overactive immune system cycling that is causing the issue there.
And I think speaking a little bit big picture, you make a pretty strong case for why preventing DNA damage is really important. But it does feel like historically the longevity field at large has not focused on this particular subfield and has focused a lot more energy on, I think, largely epigenetic reprogramming and also like lifestyle like changes like eat less, exercise. Like why do you think epigenetic reprogramming has been given the spotlight and why like this particular subfield of yours has been kind of like left to the wayside?
Genetic reprogramming is really, really exciting because it promises reversing aging. And so if you're already, kind of pushing your later years and you want to live, especially if you're motivated by living forever, then epigenetic reprogramming is one of your very few choices. I mean, there's the field of replacement that's coming out that's becoming more and more exciting, where the idea is like you just like replace body parts, which is still a very young field. But reprogramming, I think, is the most mature field that has that idea of like, okay, like maybe you can do something right now and then live forever. Like there's this whole concept of like longevity escape velocity.
Yeah.
And if you're going to hit that, you probably need something that will reverse aging, especially for the people who are already older. So that's I think the kind of biggest reason why reprogramming is exciting. In addition to a few mouse experiments that I think were really, really compelling. The biggest reason why genome instability is toughest to reach, and we think about this a lot in terms of clinical strategies, is prevention in general is hard. Prevention is expensive. Prevention takes a long time, like by definition. It is very hard to do, and most genome instability diseases are going to be inherently prevention oriented because in most cases, once the genome instability has already occurred, once either mutations or inflammation, it is not something that you can, it's already there. Like that is. Maybe you'll slow down the progression at best, but you're not going to reverse it by preventing more damage. The damage is already there, if that makes sense, right? You can kind of think about this as the simplest form is like Huntington's, right? Like if you already have the crazy high somatic expansion, if you already have a lot of copies of HTT, Huntington's is caused by expansion of a region in this gene called HTT, then preventing more expansion isn't doing anything for you because you already have all of that. And so most of the thinking around how you treat genome instability relates to prevention, and prevention is just tougher, so much tougher to do.
If I think about what the major prevention drugs are, I'm thinking like antihypertensive, statins, but even then, they like those aren't a close analog to this because they have pretty easy biomarkers to tell like quickly how well this is working or not. Is there like any like drug class in history you can point to as like what you're like would you think that like the parallel journey for permanence is going to look like?
Statins actually a great one. I mean, the most very directly, vaccines would be
Oh
the
yeah,
easiest,
yeah.
right? One of the two, there are two drugs approved for cancer prevention. One of them is Gardasil, which is the HPV vaccine.
Mm.
Cancer prevention drugs also, like there's one cancer prevention drug that exists to my knowledge, and that's tamoxifen, where they showed that tamoxifen is effective as a breast cancer treatment, but then there's a huge study that showed it can also prevent breast cancer. So the kind of a
Mm.
clinical path has already been set. Statins, GLP-1s, these are actually kind of like very similar to what we're trying to do in that they were originally approved for very narrow indications, for people who are at very high risk of a disease. And then gradually, as people showed that they were relatively safe and effective across a whole broad of different indications, they gradually label expanded. And so it's the same thing that we see here in cancer prevention, where we'll probably start out with people who are very high risk of disease and try and prevent them from getting cancer. But then slowly, as these drugs try to be safe and working in more and more people, then we'll just expand.
I'm curious. For tamoxifen, I was not aware of that. What like do we know what's going on there? Like why is it able? Like does it have like a DNA protection mechanism or is it doing something else entirely?
That's fascinating. I can speculate. So the very concrete, what's known is that tamoxifen, is modulates the estrogen receptor signaling,
Yeah.
and so that's also why it's like a drug that prevents breast cancer. I think the kind of conventional wisdom for breast cancer prevention is that you kind of have, in women, you have this kind of like idea of like a pre-cancer where it's not quite cancer-like yet, but estrogen signaling can help push it over the edge.
Mhm.
And so if you block that signaling, then you just prevent it from going over the edge into being cancer. So that's why you're preventing cancer. With that being said, like there's a lot of fascinating overlap between estrogen receptor signaling and DNA repair.
Mhm.
There are only hints of it out there. As an example of what I'm talking about, if you look at GWAS hits for age at which a woman undergoes menopause, almost every single hit is a DNA repair gene.
Hmm.
And it's strong. These are massive effect sizes. There's like one, variant that's pretty common in the GWAS hit where if you have it, you undergo menopause like a year and a half earlier. Uh, or maybe it was a year and a half later, regardless. But yeah, I mean, there's like a very strong correlation between, like, there's a link between estrogen receptor signaling and DNA repair.
Is I guess this is going a little bit like off the beaten track of like DNA repair mechanisms. But does this imply that like if you're a woman and you're above like 40 or 50, you should be on tamoxifen?
Probably not. I don't.
Okay,
To
okay.
be honest, I'm not knowledgeable
Okay,
enough in
okay.
that field, and the menopause itself is probably not. I mean, could or could not be estrogen receptor signaling mediated. I'm not sure.
Okay, okay. That makes sense.
Yeah.
I'm curious, and like related to this whole like cancer tumor thing, do you imagine that DNA protection chemicals would also help in preventing cancer from adapting to whatever therapy you throw at it? Or there's something about cancer that's a little bit weird and like these methods may not necessarily work for the cancer itself.
Yeah, I've thought about this a lot of
Mm.
like, can you prevent cancer progression?
Yeah.
And in fact, there's a very interesting study done with nicotinamide, which I mentioned earlier as a pretty good DNA repair drug,
Mm.
in terms of preventing, I want to say, secondary malignancies from a melanoma treatment.
Mhm.
And it was effective. It worked. And the patient population was like 400. It was like 400 patients each arm. The trial was like a year and a half, and they showed that sure enough, if you give patients who had once melanoma, then you have like a 30% decrease in the risk of a secondary malignancy 2 years later.
Mhm.
Um, I think it'll depend a little bit on the cancer. Like some cancers are known for being highly, they get a lot of mutations throughout the
Yeah.
course. Some cancers don't. A lot of cancers, I think it's more of a progression is more defined by immune cell exhaustion than it is by adaptation of the cancer itself.
What do you oh sorry, go ahead.
So I think it's going to depend a little bit on the specific cancer type.
What do you think? Do you think the first indication for permanence is going to be for stopping the progression of existing advanced cancer or for preventing cancer from occurring in the first place amongst some like high risk population?
Probably the latter. Yeah.
Okay. Why not the former?
The former is exciting. I mean, when I think about indication selection, I think about reasons you could fail,
Hmm.
right? And if there are patients out there who are going to get cancer and we have a drug that should prevent mutations and they're getting cancer from mutations, then it should work.
Mhm.
If we fail, then that means that our drug just didn't work, right?
Mhm.
Like that is a very clear indication. If you prevent progression, if the indication is prevention of progression, progression can happen for a lot of different reasons. And so I would hate for our drug to work really nicely and prevent mutations and then that not impact the endpoint.
That's fair.
Yeah.
That's fair. Okay. Yeah. There's a lot of reasons a patient could die for reasons unrelated to mutations. What were the other things on the table for indications other than cancer? Because you mentioned a lot of other things in the blog post.
We've thought a lot. I mean, so for example, if you look at which drugs right now are genome stabilizing or, antioxidants, for example, things that prevent damage, like to my knowledge, there are two indications right now that people are pursuing. Epsilon is a drug for ALS, and neurological diseases are very well known to have DNA damage as a driving mechanism. And so ALS was interesting to us, especially because there's already been a clinical success for an antioxidant there. Dry AMD, like currently it's an eye disease. The main treatment or like not even treatment, but like the main way that you prevent that is through this cocktail of antioxidants. And so when we think about, okay, like what is something that we can move into where we see there's already success, there's already a path, like those two stand out because it's been done before. And then from there's other indications where there's a very clear causal link between genome stability. I cited Huntington's before. Alzheimer's is looking increasingly interesting too, kidney disease, where there isn't currently beaten path, but there's genome stability is clearly related to progression or initiation of disease. Those were interesting. I think the main thing for us was mission alignment. Like our vision is to do cancer prevention and longevity. If we have a drug, what a lot of longevity companies I think run into is the way that you treat a disease ends up looking a lot different than the way that you address longevity. What I mean by that is that let's say that you have a drug that you want to put in as a cancer therapeutic, right? And you think that it also has a pro-longevity mechanism. The downside to that is that the way that you think about a cancer therapeutic is very different from a safety point of view, for example. That you can dose extremely highly if you have a cancer therapeutic because otherwise the patient will, presumably die.
Yes, yeah.
There's not something you can do for longevity. You want to have this lowest possible dose that where there's no side effects and the patient is just healthier. And so when I think about overlap with the mission of we want to do something that's pro longevity, cancer prevention is really well aligned because similar to longevity and cancer prevention, what you're trying to do is prevent a new disease from occurring. You want something extremely safe. You want something that you can take orally. You want something that you take for a very long time. You want something that has minimal drug interactions with every other drug. So in terms of just thinking about the problems that we're solving on one path versus the other, they're basically the same problem. And so we like that as a case. But of course, we could go into multiple programs. I don't think we're married to only doing cancer prevention.
Yeah. On the topic of actually taking the drug itself and it systemically going throughout your body, what do you imagine is the like if you do go like the cancer route, do you imagine like whatever your drug will be like injected at the site of the cancer? Or well, actually, I guess in the case of like you taking the drug to prevent cancer in the first place, you have this like systemic application of the therapeutic. Where do you want the therapeutic to most get into? Does that make sense? Like do you want it to most get into like hematopoietic stem cells? You most want to get into muscle cells? Like where is the primary place that you think is important, either for longevity or for cancer?
It really depends on the occasion. Like, for example, if you look at xeroderma pigmentosum patients, right? They have an extremely high risk of cancer. It's a really rare disease. That's mostly caused by sun damage. And so you really, really, really need the drug to get into the skin. For Fanconi anemia, these are patients that are mostly getting hematological disorders and a bunch of solid tumors. And so in that case, you wanted to have it get to the bone marrow, but you also kind of want to get everywhere. If you're thinking more common diseases, Lynch syndrome, that's colorectal cancer I mentioned earlier. So you need it to get into the colon. So it really depends on like the indication in question.
So to some degree, I guess when you say like the indication for Permanence Bio is cancer, it's probably going to be more specific than just like cancer. It's going to be like a very specific organ, for example.
In the beginning,
okay,
I imagine it's going to be a lot more specific. Yeah.
Gotcha. Do you think we'll ever get to the point of having this like systemic, like negative 10% reduction in cancer across like all types of cancer because of genome stabilizing drugs? or there's something about, actually, I guess this is a broader question of, are there, okay, so like some context for this question. I know that for at least epigenetic reprogramming, different cells reprogram at different rates. And so there's this weird problem is if you want to reprogram like a neuron, you need to almost like go slower in the reprogramming process versus like a muscle cell. Is there something akin to that for genome stabilizers where different cell types need like a different level of dosage?
Yeah, that's a very good question. I mean, to some extent, like from on a very, very high level, yes.
Okay.
The cells that are going to be accumulating a lot of mutations tend to be the ones that are dividing a lot. So
Mhm.
you want to get into your bone marrow, you want to get into the colon. And so from that point of view, yes. The other thing is, this is both a blessing and a curse for us. Kind of what I mentioned that we have this like cancer prevention / longevity alignment. Like one of the biggest things for us is safety.
Mhm.
And so what we need is a drug that is fine in everything. If it gets, no matter what cell type it gets into, no matter where it works, it should still work.
Mhm.
And that's very, very important. And that's why we're testing so many different cell types, because we find out that we're working in 4 cell types, but we're toxic in the 5th one. We just cannot pursue it.
Mhm. You did a lot of research on the epigenetic clocks during your PhD, but from just scrolling through your Twitter feed, you're pretty pessimistic on the overall utility that they can provide. I'd love to hear your current take, or your take on the current state of things in the field, and perhaps on biological clocks more generally.
I think I've been thinking about this a lot lately. I think clocks are very promising from a, perhaps not right now, but probably very soon, from a clinical kind of, biomarker point of view. So looking at, hey, we have this intervention, we give it to patients. What do the clocks say after 2 years? I think there's a primary little bit of challenge there in that like a lot of these clocks are difficult to interpret. But I think slowly the field is solving that problem, and their power, the clocks are getting strong enough to where they have real meaningful predictive power for functional endpoints that are just would take too long to get to using a conventional trial format. And so I'm very bullish on that front and excited to see where that goes. And that's especially helpful for prevention. When we, what would be nice is if we had a compound that, is not only cancer preventing, but actually also, longevity extending. And you can tell that with a biomarker off the blood that you trust a year into the trial. So you don't have to run the full, like, long cancer prevention trial. So from a prevention point of view, I think biomarkers and clocks are a godsend. And as basic research tools, they're also very helpful. I mean, most of my PhD was kind of trying to use epigenetic clocks to understand to what extent aging is a, kind of a cells getting old and getting not working mechanism versus the system is collapsing mechanism. The place where I think I'm a little bit more pessimistic is in like direct to consumer, like individual person takes a clock perspective. And in terms of, I'm not sure to what extent that like, it doesn't seem very actionable and it doesn't seem very accurate. A lot of the technical noise there is higher than anything that you could possibly impact. And so I think it's, unless you have the money to, buy like 100 of these things, there's not a whole lot of value that you get out of buying a epigenetic clock off the internet. And I've never done my own like tests. So I think that's where I'm pessimistic. But I think in terms of where the field is going with the biomarkers of aging consortium and looking at, clinical trials, I'm very, very excited.
How much does the biological clock you use matter based on which therapeutic you're throwing at the problem? So I imagine for like the like retro altos who are all like epigenetic people, epigenetic clocks are the most useful for them. Are they also useful to you, or are there some genome stabilizing clocks that you're interested in?
That's a cool question. I think we're getting to a point where you could actually use mutational burden as a clock.
Oh, okay. I did not know that.
Yeah, we're getting there. We're not quite there yet, but I think within the next 2 or 3 years we'll start seeing that, or something like that. I think what's most exciting are the systems clocks.
What
Like
were those?
this like basically there's I think 5 papers out now, but there's a flurry of them where so far everyone's been mostly looking at epigenetic clocks in terms of blood. You take someone's blood sample and then that tells you their epigenetic age, right? But the question is like, there's not just one dimension to aging. Like to one extent there is. Like I mean, like a 60-year-old is a 60-year-old, a 40-year-old is a 40-year-old, 20-year-old is a 20-year-old. But I mean, there are 60-year-olds out there who like have perfectly healthy lungs, but they like have a failing heart or vice versa. And so the question was, can we use epigenetic clocks or proteomics clocks to understand, to what extent someone's like at high risk of a certain type of disease for a specific tissue or organ? Those are exciting. And I think when you're thinking about, again, like diseases, those are very, very good, because let's say that you have a drug that's supposed to improve sarcopenia. Well, maybe the muscle clock is going to be the most effective at predicting how well the trial will go a year before you hit the endpoint.
Mm-hmm. Let's say you meet 100 people, all of whom are 110 years old. Do you what do you think is there a primary geroprotective mechanism that you suspect all of them have? Or is it all likely very heterogeneous amongst them?
Yes, that is a good question. I think it's going to be the one. The reason I say that, so there's this saying in the longevity field that I like a lot, and which makes me laugh, which is like, if you want to live to be 90, like, what do you do? And the answer is, oh, you should like diet and eat well and exercise and sleep and do all these healthy things. And if you want to live to 110, what do you do? It's like have parents that live to be 110.
Yeah.
And so if you look at a 110-year-old specifically, I think it is a genetic, like an improvement in genome stability. I think that like the lifespan curve, the aspect of aging that fixes your lifespan is likely mostly genome stabilizing. So that would be my prediction, but of course, like we'll see. There's some evidence to support that. Like people have found there's a variant in sirtuin-6, which is a known genome stabilizing protein. And that is enriched in people who live a very long time. But like I think there aren't even enough individuals to really have a strong enough like study to predict that.
Well, I suppose like an easy way to study this is like study like the mutational burden, like the average mutational burden of like some sample amongst these 100 people who live to be 110. Is there some reason like that is not like a trustworthy number, or is it just like we don't have enough samples?
We do not have enough samples.
Gotcha.
Yeah.
Okay. Somewhat related to this, it feels like longevity startups in general have pursued singular mechanistic hypotheses at a time. Like some groups are pursuing only like replacement, some groups are pursuing only epigenetic reprogramming. You're pursuing DNA stabilizing stuff. It has long felt to me that like aging almost wants to occur, and so if you plug in one of like the 7 holds that cause aging, the pressure on the remaining 6 holes will simply intensify. Is this like an accurate mental model of aging as a concept? And 2, do you imagine like you'll be forced to like move into other therapeutic areas for longevity, or that genome stabilizing is like a really good base to start off on?
Then the first time, my own mental model is almost the opposite.
Oh.
Where, like people have shown that, I mean, like a lot of these hallmarks of aging, these aging dysfunctions like feed off each other. And so if you remove one, you'll probably actually release the tension off a lot of the other ones. Like as an example, I mean, like you can easily like inflammation leads to dysregulated nutrient sensing, right? Because suddenly your immune cells are like eating all of the different, nutrients that other cells should be getting, which then causes mitochondrial stress. Mitochondrial stress induces genome instability, which leads to telomere attrition. And so like, if you just didn't have that inflammation in the first place, probably the other ones would be a lot better off too.
Yeah.
So yeah, I mean, the core sentiment though, I definitely share of, there are a lot of longevity companies that just like focus on the one thing. And even when we think about genome instability, I think by the time that someone's like quite older, let's say 85 plus, it's, doing genome instability alone isn't going to be enough. In fact, I'm not sure to what extent you'll see a big effect. It's kind of why we're focusing on this prevention angle, because the hope is that, early on, the primary thing that's affecting you as you age is genome instability. And so the earlier you treat that, like as an example, like inflammation is not that much of a thing when you're 25 or 35 or 45 or 55, right? It only becomes a real hallmark of aging, like past the age of 65. And so, does it make sense to prioritize genome stabilizing drugs for someone who's like 85? Probably not. At that point, I would probably hit the other hallmarks.
Yeah.
But for someone who's 30 or 40 or 50, like absolutely, that would make a huge difference.
With, again, quite like a very, very dumb question. When we talk about a cell's ability to repair its own genome, can that happen like hours after the insult has taken place? Or is it kind of like the repair mechanism is only available like a few seconds after the damage has been done, but after that the information is lost?
Oh, that's also that's very interesting. It depends. And the concrete answer is all of it.
Okay.
And what I mean by that is like there are stem cells out there, there are quiescent cells out there that get DNA damage that just sit there with the DNA damage for like months.
Mhm.
Because if they're not doing anything, that doesn't really matter.
Yeah.
But as soon as they emerge from quiescence, suddenly they turn on their DNA repair machinery. They open up their DNA, all the DNA repair proteins come in, fix all of it, so on and so forth. So versus there are certain types of, like a double strand break, you better fix immediately,
Yeah.
or the cell is going to immediately become senescent and then die, or die. So, yeah.
I guess like the more specific question I'm asking is like you say like if you're 80, we probably don't have anything to offer you. But can you not imagine that if you upregulate their DNA repair mechanisms and like allow them to go through this like rough period of like cells not dividing because they permanently think they're in a permanent repair state, and then you give them the genome stabilizing drugs, do you think there's anything there, or is it just like their cells have been through so much information loss that isn't like not recoverable?
The way I think about it is, my mental model of aging is like a boulder rolling down a hill.
Mhm.
And so, like, in the beginning, there's not a whole lot going on. The boulder's not moving very fast. And the bottom, it's like going at full speed. So, trying to recover reverse entropy is tough, inherently. I mean, that's just like a fact of nature. And like biology does have mechanisms for doing it. And that's kind of what the reprogramming field is pursuing, is this idea of like returning back to a natural state. It's just very, very hard.
Yeah.
There's definitely like some synergy. You could imagine like a reprogramming therapy plus a genome stabilizing therapy to kind of like push someone back and then keep them there, so I definitely can't say that, I think we will have things to offer for people who are very advanced in age. Plus, I mean, if you stabilize the genome, there's just certain diseases that old people, that old elderly people get that could be very helpful, like autoimmune diseases I mentioned earlier, or Alzheimer's or something. It's just much harder
Yeah.
at that stage.
Outside of genome stabilizing drugs, what is the second most area of longevity, like therapeutics, that you're most bullish on?
That's a good question. I like reprogramming, but everyone likes reprogramming. I think just in general, hematopoietic stem cell treatments excite me.
What is that exactly? Or
Like,
what are you actually doing to those cells?
let me think, what is exciting? I mentioned earlier the replacement field is very big. And so that's kind of like I'm thinking about hematopoietic stem cells. I'm like, okay, if you were to take a human body and you were to replace something in them, right? And you were to weigh the cost trade-off of like, how hard is it to replace, how difficult would the surgery be, versus the maximum possible impact that could have beneficially of rejuvenating that organ or that system. Like that's where I'm really excited about bone marrows and hematopoietic stem cells, because they're responsible for your entire immune system. They're responsible for your entire blood system. And they're very relatively small niche in a relatively small part of your body. And so if you could actually replace all of them, which is feasible, or at the very least, like theoretically very feasible, then the benefit that you would see would be huge, because you just rejuvenate your entire systemic, circulation.
Going back to this group of 110-year-olds, do you imagine that the vast majority of them have extremely detrimental mutations, somatic mutations in their hematopoietic stem cells? Or it's kind of like, if you're prone to having mutations in that area, you're probably not going to live to be 110.
It's more the second.
Okay.
So there have been studies looking at, this is time to get into deep immunology. There have been studies that have done on people who are centenarians and looking at their immune systems. And their immune systems are weird. Like, as an example, there's this like relatively rare subpopulation of cells in, younger people, or even elderly people, just everyone in general that isn't, one of these increased centenarians, called a cytotoxic CD4 T cell. And CD4 T cells are usually helper cells. So in some ways, it's like an oxymoron. It's like they have cytotoxic helper cells. They have cells that kill and also help. And because like this is a very, really rare cell type, like it's weird, it's grossly understudied. People don't know what it does. Like it probably helps against cancer is one of the theories. But it's a very understudied cell type. Turns out that there are, people who are 105 years old where something like 30% of their T cells are this bizarre, weird cell type that is like maybe 0.01% in a healthy individual.
And those people tend to live longer?
Yeah.
Huh.
Yeah, like they're enriched in these like super centenarians. So that could mean a lot of things. That could mean that they have a mutation or some genetic variation that makes them more predisposed towards having the cell type. It could mean that every other cell is just so vulnerable that they die off while this one weird cell type survives. So there could be a selection bias. It could be that as they age, they got a mutation in their hematopoietic stem cell system that led to them producing more of this very beneficial cell type. Which one of those is, it could be like a secret fourth thing. None of it's known, but yeah, the hematopoietic system does weird stuff in very old people.
That's interesting. Is there like anywhere else in the field of longevity where amongst the people who live a really long time, there's something unique going on about them biologically, and it isn't just they're better at doing the normal things that everyone can do?
Oh, that's a good question. Nothing immediately comes to mind, but I think there might be one other thing I remember reading where I was like, well, they do what?
Cool. What is currently going on inside of Permanence? What these, like, obviously we have discussed your thesis at length, but I'd love to hear about any details that you're able to share about what's going on inside.
Yeah, a lot of it's like what I've sort of shared already, like a lot of testing, a lot of screens, a lot of assays, a lot of, just looking at the battery, ordering every single best genome stabilizing thing that we could find and then putting them head to head and then doing some iteration on those and trying to improve them. Different cell types, different tissues, different types of damage, just doing the whole battery of assays. I think our first thing is just like look wide. Like what general patterns do we see? What are the most effective things? In what disease models do certain types of compounds perform the best? And then as we kind of build up a database, as we build up a knowledge base internally, then that is going to be pointing us towards like specific compounds. We've gotten some incredibly exciting results lately, where we have something proprietary that seems to be outperforming the best of the best of everything else that we've tested. So we're starting to eye mouse studies, and that is a very, very exciting thing. Especially from a cancer prevention study, because doing a cancer prevention mouse study is a lift, and it takes a long time. And so what we're doing a lot is like, what is the best model? How do we screen this? What do we look at? What do we model? What do we like? Where do we look at mutations? Do we not? What do we look? The details of how do we turn something that looks really compelling in cell assays into 3 steps closer to being a drug that we can give to patients.
When it comes to designing these DNA stabilizing mutations, like I tend to think of molecules and proteins as like they're attaching to some cellular receptor, and like one way you can conceptualize like how well is this going to work as they transfer from like a mouse to a monkey is like how structurally conserved this receptor is. Is there like an analog to that in the DNA protection world?
Not really.
Okay.
I mean, so it depends, right? So like, broadly, yes. So broadly speaking, like frequently, I kind of mentioned there was a few other genome stability companies that are looking at hitting things like DREAM or sirtuins or whatever. And so from that point of view, yes, like your guy's still going to be titrating binding. You're still going to be hopefully finding the best possible binder that is going to be attached to this particular thing. If you have something that's mopping up damage, not really. I mean, you still have to do your dose curves, or you should do your dose curves of figuring out what is the minimum dose that leads to the best improvement. But like your readouts tend to be less biophysical. Like you have enzyme plus compound, how do they bind? And they tend to be a lot more functional. You have cell, how is it reacting molecularly to damage to this compound? How is it, is it staying alive? What does it look like phenotypically, et cetera, et cetera.
When you're quantifying how well a particular molecule is working, is it kind of a binary yes or no on these functional assays, or is it a very wide spectrum?
It's a wide spectrum, yeah. I mean, like the biggest thing that we're thinking about right now in terms of assay design is like, how do we get the cleanest separation between the things that we're testing, right? Some types of damage are just going to kill everything, and some types of damage are going to do nothing, and so you can't see a difference between your compounds. But like, how do you hit that sweet spot where you can see that some things are working but others aren't?
The way that I've been conceptualizing DNA protection mechanisms is that they affect, for like a given cell, they affect the entire genome all at once. Is that a correct way of viewing it, or is it often the case that you want to protect specific segments of the genome at a certain point?
depends. I mean,
Okay.
for some, depends on the mechanism, depends on the drug. Like some really do not do anything next to histones, and they only have an effect on open areas of DNA. Other ones, vice versa. And of course, if you're improving DNA repair, then certain types of DNA repair mechanisms are more involved in certain parts of the genome than others. So I think it's going to be very molecule dependent.
I imagine, just like given what you've said about the DNA protection field, it feels like it's almost certainly relatively immature compared to most other longevity fields. As such, I imagine like Permanence as a company is an area where there's relatively little established ground for what empirically works, what theoretically works, etc. Have you learned anything that you can share about preventing DNA damage that perhaps runs a little counter to how biologists might naively think about the problem?
Yes. I will say that we're very lucky. Like in some ways, the genome stability field is nascent. In some ways, it's very old. I think the idea that DNA damage is tied to aging is something that a lot of thinkers have thought on before. And there's just an incredible, I mean, we're standing on the shoulders of giants here. I mean, looking at the number of people who have developed antioxidants in mouse models. Even, I mentioned that we're like screening the drugs that we're developing across the best in class. I mean, that's every single one of those things that we're testing against were developed by teams of people trying to find the best possible, protecting agent against something. And so there is a lot that we've learned from everything and everyone that has come before us, in the cancer prevention space, in the genome stability space, in the aging space. So yes, so there's a lot there. In terms of what is something that's counterintuitive and different that we're doing, I think it is really trying to figure out what I can say here or what I should say here. I think one of the biggest things is just like the recognition that different types of damage lead to different effects and different cell types. I think people, like, there's been a very common trend of we're going to take some fibroblasts, we're going to look at gamma H2AX, and that's just cross our fingers and hope that's going to translate to everything. And I don't think that's how it works. I think what we've learned, what our intuition was, and that has absolutely panned out in our assays, is that you need to look at a lot of different things to get a very big picture view of what is DNA damage doing and how can you prevent it.
And there's no functional readout that you can trust above everything else.
Right.
Okay. Is like somewhat on a related note, and I'm not sure if this is something you can answer, what is the most exciting internal result that you're currently seeing, and why do you consider it exciting?
Oh, I mentioned this earlier. I mean, it's just the fact that we're seeing, some proprietary compounds just work across everything. Like what I was really scared of, and this was absolutely possible, is that we would have 4 types of DNA damage, and then that we would be screening of, protecting against, and then you would have like 3 compounds that would work for 1, 3 that would work for 2, 3 that would work for 3, 3 that would work for 4, and then none of them would overlap.
Yeah.
And the fascinating thing is that we do actually see that with some of the control compounds. Like there are, I mentioned earlier that we're screening like nicotinamide, for example. We're screening astaxanthin, which is this like incredibly powerful antioxidant that algae that salmon eat produce. To my knowledge, I think it's like one of the most powerful antioxidants that has ever been known. We were screening a whole lot of different genome stabilizing compounds, and we do see that in some of them where they're really effective in like 2 out of 4 or like 3 out of 4, but like they're really harmful in the 4th one or something. And so I'm really excited to see that we have potential compounds that we want to move into mice that are helpful in everything. They're not necessarily number 1 in everything, but they're high up there in everything. And that's good.
When you're designing these molecules at Permanence, how do you like select the molecule in the first place? Like if there's no like cellular receptor that you think like I have this ligand that's binding to this one epitope, and you're in this world of like I want to bind to like this backbone, or I want to bind to the chromatin. Like what's your mental process for selecting these molecules in the first place?
A lot of things. Now I'm very much thinking about what I can say. We have a hypothesis for what makes a really good genome stabilizing drug.
Okay.
And we are like, first principles, like we are hoping that that hypothesis is correct. It is backed by literature. It is a little bit contrarian. We're testing out whether or not that hypothesis pans out in our cell screens and in other screens that we're doing. So far, it seems to. So given that it works, then the question is, can we improve upon that mechanism? And we're doing a lot of things for that. So for example, we're doing some first principles AI kind of prediction algorithms to see, given that this is our hypothesis, can we create new composition of matter that does that particular hypothesis better? And then there's a second layer of AI that we have where the idea is every single assay that we have that is finalized and every single result that we get for every single compound, we feed that all into a massive database. And then we we haven't quite gotten there yet. But the plan is in the next 3 months to go, okay, like now that we have this huge amount of data and structural data on all these families of compounds, can we actually generate something new that is, that a really powerful medicinal chemist could not have thought of? So we'll get there as well.
When it comes to Permanence's usage of AI, the way that you phrased it implies that it's like primarily LLM based and not like a molecular model. Is that correct?
We do both.
Okay. When you're using these molecular models, like what do you like are you like taking the DNA and trying to find something that binds to the DNA, or am I conceptualizing it incorrectly?
I will not tell you.
Okay.
But it's very cool.
Okay.
I wish I could. I genuinely wish I could.
I think it's helpful to think about ambitious companies in terms of the bets they're taking, because I think it's difficult to do an ambitious thing without making a bet on where the future is going. Clearly, it is known that DNA damage prevention is important and that there are mechanisms that help prevent or fix it. What do you think is the bet that Permanence is making? Or more phrased differently, if Permanence completely fails, what do you suspect the reason is?
The clinical trials will be tough. The clinical trials for cancer prevention, have been done, but they're all difficult and they all are expensive and they take a long time. The upside is massive, both in terms of good for humanity, but also in terms of, market. And there's little weird things that can go wrong. So like as an example, on one of our advisors is Professor Dr. Sir John Byrne. He's an absolutely incredible man. He has run the world's longest cancer prevention trial ever, clocking in at 20 to 25 years.
It's still ongoing?
Yes,
Okay.
actually. He ran the CAP series of trials, which were basically testing aspirin for preventing colorectal cancer in patients who have Lent syndrome. And 5 years into the trial, they wanted to look at actually preventing colorectal cancer, but they had some like secondary endpoints. And one of those was whether or not you can prevent polyps, because polyps are on the path to progression of becoming colorectal cancer. And 5 years in, they saw no decrease in polyps whatsoever. And they were really hoping that this polyp result would work because, you don't want to wait until patients get cancer because it's going to take another 5 years and, be a mess. And they almost killed the trial at that mark because they didn't see any, decrease in polyp count. They let the trial run another 15 years. And what the result was there was a 40% decrease in colorectal cancer in the patients who were taking aspirin versus the patients that weren't. And so like the bet that we're taking is that there's not going to be something ridiculous that falls out like that. Like a lot of clinical trials, just like, especially ones that go long, tend to fall apart for reasons that have very much nothing to do with the mechanism, or, being wrong on the core biology, but just something else like that. And so that's kind of where we're the most worried, I'd say.
What do you imagine the length of whatever clinical trial you run would be? Like, would it be 5 years, 10 years, even longer?
Definitely not 10. And it's got to be less than 5.
Yeah.
I mean, it's a little bit of a math problem. So like the more patients you have, inherently, the less long it takes, but the more expensive it is. So if you have an incredibly, and then, as a subset of that, the more highly cancer predisposed patients you have, also the shorter it takes and the fewer population, the fewer patients you need, right? So we're kind of, we probably don't want to run a cancer prevention trial that lasts longer than 3 years.
Yeah.
There are some cancer prevention progression trials that we can do in as little as 1 or 2. But it's going to be a long clinical trial for sure.
And I guess kind of working in the longevity field seems like an excellent way to grow pessimistic as to anything working out at all. You've posted also in the past on Twitter about how it is a shame that the ambition of the field has dramatically lessened than what it was 10, 20 years ago. How do you personally stay motivated in such an extraordinarily difficult field to work in?
It's just so worth doing.
Yeah.
Like I got into the longevity field because I used to work in the kind of late stage cancer field, and I was just dismayed at the idea that, the way that we set up our healthcare system, the way that we treat patients is that we wait until they get a very serious disease and then we hope to bring them back from it once they're at the brink. And then most of these drugs are on operating on very specific mechanisms that don't help anything else. So the promise of the longevity field that you can actually modulate something that's way upstream of everything, that you could prevent disease, and not only one disease, but nearly every disease. Like what else is worth doing?
Yeah.
I mean, as far as time immemorial, people have been having this idea of like, how do you extend lifespan? How do you, I mean, even from the point of view of mortality, but which is a little bit less my interest, but just it's the most effective revolution of healthcare that we can imagine, and I think it's worth fighting for.
I mentioned in your introduction that you did a PhD at the USC Buck Institute of Aging, which obviously an aging institute, but like walk me through like the journey leading up to getting the PhD. Like what made you realize you do want to spend at least some significant fraction of your life working on this?
Yeah, I used to work at Arsenal Bio. I think that's kind of where I got my intellectual start. Arsenal Bio being a CAR T immunotherapy company that has incredibly advanced technology in that particular space, and my job was improving screening. So the question that would like come into work every day thinking is, okay, here is a way that we can test, 10 ways of improving a CAR T to kill a cancer cell better. How do I make that 1,000 ways? And I have like 2 months. And that was a very fun problem, and it taught me a lot about the importance of the details in screening. Of like I kind of mentioned earlier that one of the biggest value adds that Permanence has is that we think about screening and testing things for genome stability in, I think, ways that are, in many ways better than other people. And that came from my time at Arsenal Bio, where I was like really enmeshed in that like screening mentality.
Mm-hmm.
I think the thing that led me to aging was, I looked at all of that and I saw, they're doing incredible work with incredible people, but could we have prevented that cancer in the first place? And there's a personal overlap there. And at the very same time that I was at Arsenal Bio, my father, was experiencing cancer, and he passed away from cancer right before I started my PhD. And he, strangely enough, he had he died from melanoma, and he had the signs of the melanoma for like a decade prior to actually becoming malignant and then, eventually killing him. And so I thought to myself, I was like, why do we wait so long? Why are we developing therapies that we can only introduce into patients once they're already, 6 months away from dying? Why can't we go more upstream? And what is more upstream than aging?
Mm-hmm.
And so I was like, I've got to do aging. When I thought about aging, and I was like, okay, where do I go? Do I start a company immediately? Do I go into VC? I realized that there was just like the aging space was very confusing, and it was like very hard for me to see like what was the signal, what was the noise, what was real, what wasn't. And it felt worth it for me to spend years of my life on a PhD to like figure that out. And so I went to the Buck, and I loved my time there.
I haven't looked too much into the Buck. Do they have a pretty dogmatic thesis on like what are the causes of aging and how to fix it? Or is it pretty freeform and they don't take any particular stance?
No, not at all. I mean, that's the beautiful thing about the Buck, and that's the beautiful thing about the PhD program. Almost every single day we would have conversations with my colleagues, in the cafeteria or something, just like chatting about like what aging is, what is the best way that you can like interface with it. We had this program called Think and Drink, where we literally just like got wine, oftentimes with like a relatively well-known person in the aging space, and just debated, lobbed questions at them, thinking about like what is aging, like how can we intervene here? What's the best thing that we can do?
Mm-hmm.
We hosted the Buck Student Aging Symposium. We had our own in-house conference where we had like speakers and like present. So like there is no particular thesis I think that the Buck has.
Mm-hmm.
They're a little bit more health span focused than lifespan focused. They lean more on how can we live better, still longer, but like on the living better aspect as opposed to like the immortality crowd.
Yeah.
But in terms of, scientifically, intellectually, like that was a place where all opinions were accepted.
Do you feel that DNA protection mechanisms were like covered well during your time at the Buck, or like that was an area that they could have like focused on more?
Oh, that's a good question.
Because like from like how you've described it
Yeah.
so far, it does feel like a relatively, even though the field has existed for a long time, it's taken a bit of a backseat to other fields. I'm not sure like does Buck give a very holistic overview of all the available approaches, or does it really focus on like what are the things that feel the most promising in like the last 5 years?
No, it's definitely much more all-encompassing.
Okay.
And I will say that DNA, like there's no like concrete genome stability lab at the Buck, for example, or
Okay.
like a genome stability center. But genome stability pops up all the time. I mean, as an example, the research that the Buck is really well known for is its work in the senescence field.
Mhm.
And of course, like how does a cell become senescent? A whole variety of things, but genome stability is one of the main drivers of senescence.
Mhm.
And so you had a lot of people, postdoc students, PIs, working on understanding genome stability in the context of senescence.
Mhm.
Ditto, well, I was working with, Dr. Eric Verdin. We're mostly focusing on immunology, but what drives a lot of immune dysfunction? What drives clonal hematopoiesis? That's mutations. So, like wherever you look, whether you're primarily focused on genome stability or not, like it always comes up.
What is a field of, and also completely feel free to not answer this. What is a subfield of longevity research or longevity therapeutic research that you don't personally think there's that much promise in?
Oh, um,
And
I-
perhaps not too much promise in is too strong of a phrase, but is the furthest away from being in the clinic.
those are two very different questions.
Yeah.
The farthest away, I mean, the farthest away, like in general, there's on the most radical side of aging, there's this idea of doing like, whole body replacement or like even BCI or,
Yeah.
like BCI, but like from an extreme angle of like you just like chop off the body and you like have a
Like
head
mind
and then,
upload. Yeah.
That's super far away. I think a lot of people are excited about that field because they come into it from like an engineering mindset. Like I think there's a very high representation of engineers who are really excited about replacement because they see it as an engineering problem.
Mhm.
But I think the reason why I think it's a long ways away is it's not an engineering problem. It's actually a biology problem that is masquerading as an engineering problem. If you talk to a doctor about how you would replace a spine,
Mhm.
that is just a whole other beast. That is not an engineering challenge. It is very difficult medically and scientifically. Not to say that we won't get there, and not to say that maybe that's like if you're thinking about the most possible efficient path towards immortality for the individuals who are immortality oriented,
Mhm.
maybe that is the most promising path, but it is a very long way away.
And what about the former question of something you don't view as very promising?
I've been bearish on. There is very little evidence that thymic involution drives aging.
I have no idea what that is. That's a brand new word to me.
Thymic involution. Okay, so the first organ to age.
Mhm.
Okay, this is also a debatable field. People will argue about what the first organ to
Yeah.
age. I will say the first organ to age is the thymus, where
Okay.
you start to see the thymus falling apart even as early at the age of 5.
Hmm.
And the thymus is really, really important. The thymus is where all of your naive T cells spawn from. And so the thinking goes like if you run out of naive T cells and you don't have the organ that creates naive T cells, then that's clearly going to negatively impact immune function.
Yeah.
The downside is that there's, been studies that have been done on patients who have had a thymus removed early, even as early as 5 years old. And it doesn't seem to really impact aging a whole lot. And I think the reason why is that you make a lot of T cells and humans are pretty big. And so as a result, like you have enough T cells to last you past where you would naturally die. I've once chatted with someone and he said like, oh, thymic involution would be a serious problem if we live to 150. And there are a number of problems like that in aging where it's like if we solved aging up to 150, then you suddenly get a whole new suite of problems.
Yeah.
And it seems like thymic involution's probably like thymic degeneration is one of them. And so I'm less excited about ways to bring the thymus back.
Purely because it's like a problem. It's like an overpopulation on Mars problem. It's not something you expect to be an issue for a very long time.
That's what the literature suggests to me.
Mhm.
I mean, of course, like the contrarian argument is that like having more naive T cells is always better. So like if you had more new naive T cells as opposed to old naive T cells, then that would be beneficial. But naive T cells also don't age a whole lot. So I've had a hard time with that. Yeah.
This is interesting because I've long wondered, like let's say we solve all 6 to 7 hallmarks of aging, we get to the 110 mark. Thymic degradation is the first thing I've heard of as like that's where it really starts to be a problem. Is there something else that is like an end game boss with regards to aging that only rears its head long past or like not usually natural lifespan?
Hmm. In the immune space, that's the only one I can think of. I'm trying to see if there's anything else that also comes up, and nothing immediately comes to mind. Um,
So
I mean,
like,
I think that the more time goes on, the more the extracellular matrix plays a role,
mm,
because like you just have a lot of extracellular matrix proteins that have a certain half life, and that half life is measured in many decades.
yeah.
Um,
Do those turn over?
that's the thing is that I mean, it's complicated. So like a lot of them do, but they when they get turned over and get replaced, they get replaced poorly.
Oh.
And so like over time, you see like problems with elastin. That just I mean, but like the problem is that it's not even an in-game boss, because you start seeing these problems even when you get like 80, 90,
Okay.
70, 60, right? Like we wrinkle, and that's a lot of that is ECM problems. So
Mhm.
like I wouldn't say that's an in-game boss, but it becomes increasingly hard to deal with, I think, the older we get.
Do you imagine the I imagine stuff like epigenetic reprogramming and DNA protection can only go so far, and like the final longevity treatment will be replacement. Is that is that how people in the longevity field generally think about it, or there are people who are like full on like epigenetic reprogramming is all you need, genome stabilizing is all you need, and if you have this in sufficient quantities, you will live to be 200, 300.
Almost everyone thinks it's going to be a combination of things.
Okay.
Yeah, I think it also depends on the age. Like, you know, again, if you're super, super old, or, you know, if you're pushing your 90s, then probably replacement slash reprogramming is the way to go. If you're in your 20s, then you're better off preventing aging and doing genome stability work. So there's a lot of like ifs involved in there.
You've mentioned in the past that DNA, you decided on DNA damage prevention relatively early in your career. I think it was something you happened upon while you were at the Buck Institute. Was there some less discussed approach to longevity that you were really wondering about and like thinking that you should start a company in? Or was DNA damage prevention the main thing you were curious about?
But in my mind, the way that I kind of like broke this problem down was, you know, as a health span or a lifespan problem.
Mhm.
Like if I was trying to optimize for how do we make people live better longer, but do you emphasize to live better longer or live better longer?
Mhm.
And genome stability is harder, but I think it'll do both, and it also will extend maximum lifespan. If we're thinking purely the health span angle, I mean, I'm just like very excited about like targeting inflammation. Like BioAge has an NLRP3 drug that I'm very, very excited by. I think that one of the primary reasons why people feel bad as they age is because of chronic inflammation. And if there's one kind of lever that I would pull to address that, it would be like that inflammation side, especially innate
Mhm.
inflammation.
What would an alternative universe Alan have worked on that is perhaps not even in the longevity space at all? Is there such a thing?
There is. Conceptually, I love oncolytic therapies.
Okay.
Um, because this is coming back from my like CAR T world
Mhm.
background, where I love CAR Ts. Again, Arsenal Bio was a great
Mhm.
time, but like you always have this antigen escape problem, right? Where if you train a T cell to recognize a single thing on a cancer cell and then kill it, then the cancer can just not make that thing.
Yeah.
And then the CAR T is not particularly effective. But there's the concept of oncolytic viruses, for everyone who's familiar, is that you just like you take a virus or a bacteria and you put it inside of a tumor, and that leads to a number of things happening. One is that you kill the tumor itself using your virus, but the other thing is that your immune system sees the threat and it wakes up. It turns a cold tumor into a hot tumor, because suddenly it sees not only is there like a bacteria or a virus that's attacking the tumor cell, but suddenly there's also like a tumor cell, and it only starts to recognize that once there's other immune signaling threats happening there.
Hmm.
And so as a more generalized cancer therapy, I'm like incredibly excited about that field, even though like clinically it's been a little bit of a mess.
Why I have heard a lot about this particular side for the of oncolytic therapies. Why do you think it has been such a mess?
It's just so hard to you're toggling three things at the same time. If you have a virus that is too good, that is incredibly effective at killing the cancer cells, but then isn't get recognized by the immune system, then the tumor can like kind of adapt to it, and then the immune system never like gets turned on, and the virus slowly dies. Or you deal with your primary malignancy, but you don't do anything with the secondary malignancies because the immune system doesn't recognize the threat. If you have a virus that isn't strong enough and is too safe, then you don't have an effective therapy. If the virus is very effective and not safe, then you start seeing like actual side effects in patients where you not only have cancer, but you now have a very serious infection, which is also not good because cancer patients have notoriously not good immune systems. And so dialing all of those to be the right level of safe, effective in terms of turning on the immune system, but also effective enough in terms of actually killing the tumor cells has proven to be very difficult. And I think a lot of companies have worked on this, and it's still a very promising field, but it's going to take some iterating, I think.
I'd like to talk to you about your process of raising money for Permanence. Permanence sits in this place of not having a clear parallel to any existing therapeutic that's out there. And at the same time, I also see a lot of VCs as saying that they want new targets, but the people who are developing new targets often struggle with raising money. I'd like to hear about your fundraising journey and whether that was an issue for you.
Yeah, I think there's a little bit of a difference between tech world and biotech world in this regard, where in biotech world, I mean, people have tweeted about this all the time and post on LinkedIn of like, look how many PD-1, drugs there are now. Look how many like TIGITs and, CTLA-4s. And because biotech VCs, especially on the East Coast, really care about validation. Like they want to have something that they trust will work, simply because every stage of the kind of like biotech, biopharma process is so expensive. So you want to de-risk as much as you can, especially from the beginning, going in. And so it's difficult to go to a biotech VC and fundraise on something very fundamentally new, because it's just like it's technical risk on top of personal risk. For tech VCs, the way that tech has been so successful isn't, you hear about the power law a lot. The idea is 95% of things will fail, 5% of things will be incredibly successful and become unicorns, and you really only need to bet on, like, even if you have a VC fund and you bet on 99 things that are 0x and go to 0, but you bet on one thing that is a 1,000 to a 10,000x, then you've like returned your fund 10 times over, so you're very happy. And so the kind of appetite towards risk is extremely different in the tech world. And so I've gotten to be, biotech VCs have been incredibly helpful and were very useful in terms of, hey, look, here are the things that we would want to see when we invest eventually. But early on, it was the tech world that was the most helpful to me, because what they saw was, hey, there's a man's trying to prevent cancer. There's a 95% chance it's not going to work because it's a very difficult journey, and that's entirely valid. But there's a 5% chance that it succeeds and you cure all of cancer, or you prevent all of cancer. And that is going to be massive. And so it's worth investing in. So I think kind of like leaning into that was very helpful for me. And it was kind of like how I managed to fundraise.
The way that I've heard one tech VC actually describe the biotech VC world is that the managing partner at these firms looks at what the top 20 pharma, like chief of sciences, are interested in, and just as like aggressive M&A to try and find the people to invest in who will appeal to one of those 20. Do you imagine that whatever Permanence produces will like eventually be acquired by a big pharma, or it'll like you'll like take this drug to like the finish line?
That's a good question. We'll see. The way that I've always presented this both to investors especially is, there's different paths that Permanence Bio could take. Like our dream is cancer prevention. Our dream is longevity too. Like prevent disease as early as possible. And at this stage, like that is a hard sell, and I am contrarian enough to try my best to do it. And it seems like we have some early success, and hopefully that success continues. So long as we can see a success moving into cancer prevention, like we want to be the #1 cancer prevention company. There is a world out there in which genome stabilizing drugs turn out to be very effective for a whole variety of different diseases. I mentioned dry AMD and maybe ALS. But the cancer prevention thing is just too much of a lift. And in that case, like we'll have a very effective drug against insert acute indication here. Maybe we'll get acquired, and that can be more of the strategy. But I think so long as cancer prevention is on the table, like we want to do it.
When you were raising for Permanence, how difficult was it to tell the story?
At first, very.
Mhm,
That was there was a huge shift between how the narrative played out. Like,
mhm,
I mean, the challenging aspect was that I came from a PhD for 3 years, and then did a VC for a very brief period of time, and then dove right into building a company.
mhm,
And that PhD mentality, like even the kind of like conversation we're having now, like is hard to unwind,
mhm,
and you have to unwind it when you're trying to raise for a company, especially among like tech investors. Um, so I remember my first pitches, like the first narratives I had was like, oh, like aging and genome instability is really important in aging. And if you can prevent genome instability, then you can prevent other forms of hallmarks of aging that then lead to other diseases. Um, and boy, like everyone was bored immediately. And so what I pivoted into instead was the kind of like focusing on the cancer prevention aspect because people really thought that was interesting and different and unique.
mhm,
And it had a huge potential upside. And so the narrative became much more into like cancer is a function of mutations,
mhm,
and inflammation. No one's targeting that mutations aspect. We can,
mhm.
like we know how. So let's just do that. Let's build drugs that prevent mutations, and that would prevent all of cancer. And that was a really compelling story.
Was it a matter of removing the science and like not explaining the exact specifics of it? Like how important was that? Like how much did like tech investors like practically care that you're doing like one modality versus another modality?
It's both. I think
Okay.
it's both making the vision grander.
Mhm,
It seems weird, right? Because cancer prevention is theoretically like a less grand vision than longevity, but the longevity means a lot of things to a lot of people.
mhm. It's kind of a squishy definition.
It
It's
kind
like
of
you can put whatever you want inside. If it was cancer, it's a little bit more definite.
Yeah,
Yeah.
cancer prevention specifically is like a much more like big goal.
Yeah.
Right, it feels like much more of a defined big goal. And then yeah, like taking the science down a notch from genome instability. Like, I mean, even the first 10 minutes of our conversation today was like, what do you mean by genome instability?
Mhm,
It's this whole thing. But instead just like mutations,
mhm.
right? And so like focusing on mutations because people know what that is and everyone can agree on a definition of what a mutation is. I think that grounded it for a lot of folks.
Why do you think the existing cancer prevention trials or like the past ones have failed?
Really depends on which ones. So a lot of them were just like kind of like there's a very long cancer prevention history, and to be honest, I'm only familiar with some of it. But for example, people very early on realized that aspirin is very effective at being a chemo preventative agent.
Mhm,
And so big pharma also realized that aspirin, seems to prevent cancer by pretty significant margins, specifically colorectal cancer. Aspirin has a few less than ideal side effects, especially at high doses. And so what a lot of big pharma companies did was they were like, okay, let's take aspirin and then improve its targeting. I want to say it's COX-1 or COX-2 targeting, but like one of the two specifically, and then improve that angle so to make it a safe thing and then really target that one mechanism. And then that will be the best ever cancer prevention drug. That will be aspirin.
mhm.
The sad thing is, and this is still, I think, a very like controversial, there isn't a clear answer thing, but from reading the literature deeply, it seems like aspirin prevents cancer through a weird off-target mechanism. And so pharma having spent hundreds of millions, possibly even billions of dollars into cancer prevention, improving these drugs for a mechanism that was incorrect,
Mhm,
like the appetite just died after that
mhm,
because, there was a lot of investment and a lot of hype and a bunch of failure, but they're just going after the wrong thing.
mhm.
Um, so I think that's part of the reason.
Do you think what like when was that?
Early 2000s, yeah.
Early 2000s. Do you think we're like coming back into the cycle of people being really interested in cancer prevention, or it's still a little bit outside of the Overton window?
It's coming back. I mean, I want to say the first ever cancer prevention conference was hosted a
Mm,
year ago. Now it's the second one. Is actually happening, I think possibly right now,
okay.
in the UK. Um, More and more people are thinking about it. I mean, the big boon here is that we're getting like the technologies to measure genome instability are coming online. And of course, there's Alex Kagan's big paper looking at aging and somatic mutations with aging and showing there's a crazy strong correlation between like how quickly a species gets mutations and its lifespan. And that technology just didn't exist
Mhm.
even, 5 or 10 years ago. And so the fact that the tools are coming online to develop these drugs, to measure these things, the biomarkers in the blood exist now. I think that's bringing a lot of fresh air into cancer prevention.
Permanence Bio, I think, just hired its first person, at least according to the LinkedIn post that you posted a job posting. Last I checked it seems to be offline. I assumed you hired someone. If so, congratulations. Um, I'm curious, like, what were you looking for in like a founding scientist, founding like first few employees?
Details. The main thing that the main thing that is challenging about cancer prevention and genome stability, I would say, is kind of, from the first part of conversation, like, how do you set up these assays? How do you measure mutations? And that is if you've looked at like a whole genome sequencing, specifically looking at DNA mutations pipeline, it is complicated. And every single little like enzyme that you use, the steps that you use, what read depth you analyze it, like how do you, like all of that matters so much. And so, like our first hire was Carlos Galicia, who I knew from my PhD, smartest guy that I knew in the PhD program. And I felt very lucky that he offered to join. But he's got such an eye for these like particular details. Like he knows exactly like, this experiment failed. I'm going to test these 4 things and that's going to make it work. So detail orientation, I think, helped a lot.
What do you think leads to that? Is it just extremely high conscientiousness, a lot of past experience, very high fluid intelligence, or like something else entirely?
Obsession.
Obsession. Okay. Well, like obsession with getting things correct, obsession with the field as a whole, or what is it? What is it exactly?
It's more of that like engineering, I think, obsession of like, if you like, it's like how do you solve a puzzle type of obsession, right? Like how do you like troubleshoot this thing and make it work obsession?
Do you think there are such thing as 10x wet lab biologists?
Yes.
Okay.
Yes. Easily. Yes. Absolutely.
And you think it comes down to this detail-oriented approach?
I think it comes down to probably 2 things. One is the detail orientation. I think that helps a lot. There's a lot of scientists who can spend a lot of time and effort on something that, because of the details it falls, but it's also about asking the right questions. I think probably asking the right questions is the more important part of that. It's so easy to spend, especially because biology just takes so long, it's so easy to spend your time for months, if not years, beating your head against the wall for something that doesn't even, like in the end game, doesn't really matter. And so the people who I think have been incredible scientists are the ones who see a gap that they can solve and they figure out how to solve it.
What does your information diet look like? Because if I scroll through your Twitter page, you often post links to papers that are completely unrelated to the field that you're working in, but you find some way to connect it, and you say like, hey, this is like super relevant for DNA protection mechanisms. Do you spend relatively little time reading papers in your own immediate field and instead are searching for like quote unquote alpha in other fields, or is it a mix?
It's a mix. I mean, I think this is also more of also a deeply personal thing, in that my philosophy towards like life in general is to surround myself with the things that I like. And to a large extent, we are a product of our environment. And so to what extent can you surround what you see and what you experience with what the kind of person that you want to be? And my Twitter is actually a really good example of that. Like I follow a relatively small number of people, and for everyone that I follow, all of them talk science. Like that is their main thing. And there are a number of people that like I would feel awkward not following because they're like, like a close friend or something, and they don't post. In which case, like every single person that I have there that doesn't post on science, I mute. So like my Twitter feed, and I never go into this For You tab. I only go into the following, like the people you follow tab. And so when I scroll Twitter, I only see like scientific papers, scientific discussions, maybe some like founders and some like now kind of like the more oriented in that direction, like biopharma, like discussions as well. But just like surrounding myself with that knowledge. With regards to papers, like I have Google Scholar alerts for cancer prevention. I have Google Scholar alerts for lifespan, DNA repair, kind of everything that I'm interested in there. And also try and surround myself with people who think about these things a lot. So like I have I'm part of a genome stability journal club group chat on WhatsApp where it's just like 30 people who are all obsessed with genome instability, and we meet once a week to like discuss some paper. We literally had that today. And so I spent an hour like debating whether or not mitochondrial DNA mutations are a causal driver of aging and what's the evidence for that right now. A lot of my other communities are also oriented in a similar direction. So it's just, like nonstop input stream of relevant information.
Do you like, I know at least one person who like in the pure machine learning world, he says like a lot of like the best papers of today are worth reading, but the best papers of like 30 to 40 years ago are also worth reading and no one is reading those. Is there something akin to that also in your space where you like read papers from the 90s and early, or I guess like 80s and 90s, or are they not super relevant?
They're very relevant, but I think much more from like a lot of I found a lot of joy in reading old like theory of biology or theories of aging papers. I've been trying to dive into those less now that I'm very much like heads down operational. But still, like, thinking about how people saw things big picture is pretty important. Especially I think as we go into I'm gonna start to sound very old now, but like as we go into a world where everything is like sped up,
Yeah.
going back to a time when people thought very big picture, I think is helpful.
You went from 3 years of doing research in your PhD, and I imagine, oh, like also your time at Arsenal, and also I imagine you did some research in your undergrad. And I imagine your day to day is not too much like bench work research, and is more being a CEO. What's that transition been like?
It's both. It's the best of both worlds.
Okay.
I mean, my day to day is I come into work at 9 a.m.
Mhm.
and then I take my meetings from 9 a.m. until 1 p.m. and then I put on a lab coat from
Okay.
1:30 until 10 p.m. and then I do science.
Okay.
So it's both actually. It's a beautiful mix of both.
Mhm.
Um, and I think it's like both things that I feel very comfortable in. Like I love operating and I love science. And at least for this period in Permanence Bio's history, which I think would probably not last a whole lot longer, the best use of my time, the best, most alpha I can bring to organization is in my scientific knowledge.
Mhm.
And so right now, like that, I'm still harnessing that. I imagine 2 years from now, that's going to be much less the case, and it's going to be mostly CEO hat. But
Yeah. You guys are roughly 8 months old, I think. Is that correct?
incorporated in October, so oh boy, yeah,
Yeah.
8 months,
8 months.
yeah.
At some point, I imagine you're going to have to make a decision of whether you want to be the full-time operator versus like full-time scientist or like full-time CSO of the company. Do you know if you're leaning one way or another?
Um, I think much more the CEO hat than a CSO hat. I think I'm very good at vision,
Mhm.
like connecting the dots between we have, a vial of 100 micromolar of some compound that we want to test in some assay,
Mhm.
and seeing how the dots connect towards how can we prevent cancer in the general human population. I think that is kind of, you know, exec coaches like to say about like the zone of genius, right? And
Mhm.
like the zone of genius that I think I have is I can connect to those dots. And I think that's very hard for a lot of other folks.
Mhm.
I'm a pretty good scientist, but I wouldn't even, like I think I can there's a lot of very, very, very good scientists out there, and who are much better than I, that I would love to hire and bring on, and so I don't think I'm as needed there.
That makes sense. Do you imagine like big picture view? And I think this is the last question that I'm going to ask. Over the next 10 years, let's say, whatever Permanence is working on works really well for a specific subtype of cancer. Do you imagine you'll keep poking away at improving, like moving whatever therapies you have into other subtypes of cancer, or would you switch to a different indication entirely?
We'll follow the data. We'll
Yeah. Okay.
see,
Yeah, that makes
right?
sense. That makes
Like if
sense.
if their assays show that there's really powerful in some acute disease model, then we've got to do that. But if it doesn't, then yeah, just keep moving into cancer.
Okay. Cool. Thank you for coming onto the show, Alan.
Thank you so much. This was a lot of fun. This is hours of yapping on my favorite topic. So thank you so much.